And thank you for the previous speakers. I'm going to be giving one more chapter in the Density Management and Riparian Buffer Study story, and Paul Anderson gave a great introduction for what I'll be talking about today. And partly what I'll be talking about today, I really appreciate Bob Danahy's presentation because he did a great job characterizing small streams, which will be part of what I'm trying to do as well. So here we go, another chapter in our riparian buffer story. The density management study was conceived in 1993 and 94, right during FEMAT and the development of the Northwest Forest Plan. And it was originated as a study of effects of thinning to try to accelerate the development of late successional and old growth upland conditions. As they were looking across the BLM landscape in Western Oregon, they saw that most of the potential sites for implementation of the density management part of the study had streams. And of course, we know now that up to 80% of headwater areas, or these small streams, can comprise up to 80% of the stream networks. And I think it must have been a hallway conversation between John Tapner and Jim Siddell in the Pink Tower during the Northwest Forest Plan summer that they had a conversation and said, well, why don't you just add a buffer study to the density management study and we can hit both of these problems at the same time. So that's what trickled down to me, and so some of the questions that were beginning at that time as the interim riparian reserves for the Northwest Forest Plan were just beginning to be thought of, and they were being implemented on all streams, the non-fish bearing streams in particular. Lots of questions were coming up. Well, what is it about these headwaters? Do we need buffers on headwaters? What are the values there? And, well, how wide do the buffers need to be? There were lots of questions and not very many answers at that time. And even as I was reflecting, as Bob Danahy was talking a few minutes ago, at the end, he gave references for the work that he and his colleagues have been doing to characterize headwaters. Most of those references are 2007 and even more recently to 2011. So we're only beginning to understand what's happening in small streams right now. These are the questions that we had at the beginning of the study, and we've been making some headway. So in the riparian buffer study, really we were out initially to start to characterize, well, what's going on in small streams? And then secondarily, let's look at some buffer widths and look at treatment effects on some of the things that we think are values, either habitat or species in these areas. So this is what I'm going to talk about today. Just very briefly, I'm going to go through some of the highlights of what we've been finding in these small streams. I'm not going to go through the details of what I've been finding, but if you're interested in details, I have lots of details with me, so catch me at lunch. So for the density management and riparian buffer study, we initially started with 13 sites, and 3 of these were on Forest Service lands, and the rest were on BLM lands. As time has gone on, some of these sites have dropped out of our analyses for different reasons, either implementation considerations, or, well, whatever, something happened along the way. But I will be talking about some pre-harvest data that we collected starting in '94 and the first years of the study, and then post-harvest data from in-stream sampling in years 1 and 2 post-harvest, years 5 post-harvest, and years 10 post-harvest. And there are a couple of side projects that went on outside of streams in uplands on amphibians, and if I have time, I'll hit those as well, but if I don't have time, that's after my conclusion slide. So the first question, characterizing headwaters, and I'm only going to hit on 2 parts of what we've done to characterize headwaters. First, we've noticed that headwater streams are much smaller, of course, and they are comprised of a mix of perennial streams and intermittent streams. And by intermittent, I mean spatially discontinuous. So the water flows on the surface, oftentimes, and probably most often in our reaches, coming out from a seep, just like Bob Danahy had explained. But then the water goes subsurface along the way. So that is intermittent or spatially discontinuous. What it looks like is that these streams have been highly disturbed from past activities, and there's side slope slippage, and that slippage goes into the stream and the water just bubbles underneath. So you get essentially hyporheic flow as you get side slope slippage or slumping into the stream. This slide shows the frequency distribution of different stream types. I call them hydrotypes. Perennial streams that flow on the surface in the spring and the summer are type 1. If the streams go into a summer intermittent pattern where they're spatially discontinuous in the summertime, but then they're continuous in the springtime, that's type 2, summer intermittent. The most frequent wet channel that we've seen in our density management sites is this always intermittent pattern, that it's spatially discontinuous in the spring and spatially discontinuous in the summer. Initially, as we were characterizing streams, we made no assumptions about what a stream was, and so we did do some characterization of channels that were always dry, and that was that last stream reach type number 7 here. So characterizing these uppermost headwaters, there's some variation in stream flow. We do have perennial streams, but quite a few of them are spatially intermittent or discontinuous. And these patterns of hydrological types are tightly associated with the amphibian and fish communities that we see out there in the headwaters. So the second part of characterizing headwater streams that we did initially with our 13 study sites was to look at the communities of vertebrates that occur in the streams. And we found that there was a community that was associated with the intermittent streams, and that was different than the community associated with the perennial streams. We also look at stream banks, and the stream bank community is an intersection of those intermittent and perennial stream animals, as well as organisms that just occur along the banks, and other types of organisms that seem to be coming in from other places, upland-associated amphibians and pond-breeding amphibians. So the banks are sort of an intersection point of a lot of different communities. Torrent salamanders, I've noted there, they are an organism that we seem to see quite frequently in the spatially intermittent streams. So they are not restricted to those intermittent streams, but they characterize those intermittent streams. They're found in seeps and small streams throughout the west side, and they're highly associated with these headwater habitats. In terms of looking at the habitat characteristics, we see that habitat seems fairly compressed in headwaters for these organisms, meaning that as we're doing our sampling, we sample primarily within 2 meters of a stream for bank communities. And that is where these animals are occurring, very tightly associated with the streams. That stream bank is wet and moist and is very rich habitat for this complex of different communities in headwaters. There's a lot more detail in our papers, and so if you would like to know more about characterizing or how we have characterized headwater streams, let me know or I can get you those papers. We also see fish. I want to mention fish almost as an aside. We see sculpins and cutthroat trout at some of our stream reaches. We've been really challenged to do analyses of the fish in our sampling because they're quite variable in their occupancy among reaches and among sites. And so I'm not going to be talking about analyses of our fish data today, but if you're interested in what's happening with the fish story, come talk to me later. The variable occupancy is something that's challenging even with the amphibian part of our communities, that we have quite a bit of variability in occupancy among reaches, even within a study site. And that compromises our ability to do some analyses with even the amphibian data. The other thing with the amphibian data, a lot of the species are occurring in very low numbers. So our low sample sizes is another challenge to analyses. And you'll see that as we go along, our sample size increases as we get more data over the years. And so as we've gone through time, we've been able to do more sophisticated analyses just because our total sample size increases through the accumulation of data among years. So our treatments. To review, we have 4 buffer widths with thinning outside the buffers. Because of the geometry of streams at the density management sites, we have not been able to look at all the different upland thinning treatments with all the buffers. In the design of the study, we tried to concentrate the buffers within one of the upland thinning treatments, and that was the moderate treatment. That treatment took the standing trees down to 80 trees per acre. And so we tried to wedge as many of the riparian buffer treatments into the moderate upland treatment as we could. But it really depended on the geometry of streams occurring at the site. And we also needed to have control streams, so streams within the untreated reference unit. So an example of a layout is here at Green Peak. We've seen this slide already today. And we were not able to get the 2 tree height buffer into this study site, but we were able to wedge in the 1 tree, a variable width buffer, and a streamside retention buffer, and have control reaches for streams in the untreated reference stand. So that's kind of a familiar theme for all the study sites. It was very difficult to find enough space to have the 2 tree height buffer in a headwater. The 2 tree height buffer would go up and over the ridge line. We really tried to have at least 200 feet of thinned area outside the buffer before we hit the ridge line. So that was a constraint, and we don't have as many replications of the 2 tree height buffer. Here's a roadmap of the analyses that we've been doing so far. In the pre-treatment characterization, I talked about that already, very briefly. Now I'll go through some post-treatment effects, only on the amphibians, in 1 and 2 years post-harvest, year 5 post-harvest, and year 10 post-harvest. And this is the template for the treatment effects, and so I'm sorry, I'm not going to show you the statistics. I'm just giving you the bottom line of what has happened in these analyses. So first with years 1 and 2 post-harvest, data from 11 sites and 68 reaches as we analyzed the bank data. But in this analysis, we excluded reaches that we did electroshocking in stream, and so we only analyzed data that was hand sampled in the stream, rubble roused, and that was 54 reaches in stream, using Kruskal-Wallis one-way ANOVA tests. The results were no negative effects on animal abundances, relative abundances in the reaches. Along the banks, we did see that newts increased in numbers in the buffers, and they declined in the reference streams. Okay, newts are a pond-breeding amphibian, and so they're one of those incidental organisms that are occurring along the streams, and they're breeding somewhere else, where they're not breeding on our sites. In-stream, giant salamanders, Pacific giant salamanders, or I guess they're called coastal giant salamanders now, they increased in the buffers and they declined in the reference streams. So that was not a negative effect, that was a positive effect. And we found no treatment effects on habitat elements. Several caveats need to be mentioned here, and these caveats pertain to the next two slides as well. Detectability is very difficult of amphibians in these type of stream habitats. However, in-stream animal detectability we've found to be very, very high. So we're fairly confident that if an animal is in the stream, we are able to census that, with the exception of the fish, which I'm not talking about right now. We do have low sample sizes, and we have uneven occupancy among reaches and sites, and that does constrain the use of some statistics and the power of our analyses. So low sample sizes may be why we're not getting some treatment effects statistically. So anyway, years 1 and 2, post-harvest, we're seeing no negative effects of the buffers with thinning. Let's go on to year 5. Year 5, 11 sites, 68 reaches. So in this analysis, we have included the reaches that we've done electroshocking along with the hand sampling, but we've put in methodology of our sampling in stream as a covariate. The main results, let's go down to number 3, using the Wilcoxon rank sum test on the bank, Western redback salamanders decreased compared to pre-treatment. Using a different analytical technique, again, Western redback salamanders and newts this time decreased compared to pre-treatment. Using a third analytical technique, randomization, which is similar to what Mark Maleeson talked about, where you do many iterations of a model in a time period. Along the bank, in that analysis, torrent salamanders decreased in the streamside retention buffer and in the wide buffer. That was on the bank. In stream, coastal giant salamanders increased in the wide buffer, and tailed frogs also increased in variable width buffer and the wide buffer. In these analyses, what we did is we merged the 1 tree height buffer and the 2 tree height buffer together into an analysis of wide buffers. In this analysis, we also redid the analysis for the 1 and 2 year post-treatment data. And I can tell you about those results later. They're highlighted here if you read really fast, but I'm going to change the slide. 10 years post-harvest. 8 sites, so we're reduced in number of sites. For the most part, the Forest Service sites have slipped out of the analysis at this time, 45 reaches. The main results are showing that for all animals, the variable width and streamside retention buffers had a significant effect with a negative coefficient, meaning a negative effect on all species. Dunn's salamander also, in particular, had a negative effect. That's on the bank. In stream, for all animals, we had a very complicated model, and we can't tell if it's a positive or a negative effect because of the complications of the model. Also, coastal giant salamanders had significant effects, but it was a complicated model, and we can't tell if it's a positive or negative. Some of the complications of this model is that we included the search method in the model. Pre-treatment count was very significant. What was there before affected what happened later. The stream width was a significant factor in the model, and the interactions among all these things were always significant. So it complicates, and we can't really tell if it's a positive or a negative effect. So if you want details on that, I can tell you, but I'm just, in terms of time, I'll go on. I'm going to skip this slide because it's too complicated. So in summary, what we're finding is that there are multiple assemblages of animals in these headwaters. The treatment effects appear to be mixed. There's not a signal that's consistent coming through these analyses, especially through the different years. Some patterns have emerged, however, and we're following up on those patterns. Overall, what we're finding is that the animal species are retained at the sites, even with the narrowest buffers. And our analyses are ongoing, and phase 2 is ongoing. As Paul explained, we're looking at an additional thinning entry, taking the TPA down to 30 trees per acre. And in some case studies, we're doing thin-through buffers, essentially no buffer. And we're going to look at that in some case study examples. So with that, I'm just going to show a slide of the case studies of the upland terrestrial salamander studies that have been done. And if you're interested, you can read that, or you can go to lunch. So thank you, everybody that has been involved with this.